On last class, we went through, excuse me, I missed you guys a long time. We had Monday off. Oh, man. We went through the process of DNA replication, okay? And I said that it was remarkably accurate, and that is a function of the DNA polymerase, right, enzyme that reads the template and incorporates the right nucleotide depending upon what Chargaff's rules of complementarity, right? A to T, C to G. It is remarkably accurate, however, mistakes do occur, okay? And so there are a fail-safe quality control methods that the cell employs to make sure that any mistakes that do happen are corrected, all right? And so there's DNA polymerases have a proofreading property or proofreading activity. They can go back and they can replace mismatched nucleotides. Okay? There are other ways where repair occurs. In mismatched repair, so where there's an A to a C or a G to a T, right? What happens? What happens when that occurs? When you have two purines or two pyrimidines there? What's going to happen to the diameter of DNA? It's not going to be the right fit, right? It's not going to be that 10 nanometers or whatever it is. It's either going to be too big or too small. That's a change in the structure. And that is recognized by proteins, DNA polymerases. They recognize that change, and they can replace it with the correct nucleotide. All right? Now, mismatch repair has other enzymes that will recognize what it is, and we'll go through what like a thymine dimer is. All right? Oh, there's also hereditary defects that occur in this. Right? Has anyone ever heard of xeroderma pigmentosum? It's a genetic defect where people can't go out in the sun because they lack this mismatch repair enzyme, right? Proteins that will correct DNA mismatches. So in some of these mismatches, you may have DNA that looks like this. One strand, another strand. Okay? How many of you like to go out like the beach? I love the beach. Okay? All right. How many of you wear sunscreen at the beach? Not. How many of you go tanning? Okay. It's okay. When you expose your skin is exposed to UV radiation. When you go outside, right? UV light, UVA, UVB rays. What happens sometimes is that your DNA actually is altered, okay? And you get the formation of what we call thymine dimers. So a thymine and a thymine, all right? So now, within that strain of DNA, you have a thymine that is linked to another thymine. That's not normal, right? So a protein will go by and recognize that thymine dimer. That's going to change the diameter of DNA, and a protein will recognize that. UV radiation also synthesizes or stimulates the synthesis of this protein. Okay? And so that protein's produced, and it will recognize the thymine dimer, cut out the thymine, right? Cut out one of them and replace it with an adenine. Right? So that's the process. So I mean, we all don't get skin cancer from getting one sunburn, right? We have ways of correcting these problems, all right? But when someone who has xeroderma pigmentosum has a genetic defect that protein that recognizes these dimers isn't produced. It's not working. So what happens to their ability to repair these dimers? Is it reduced? Yes. So you can't clear the dimer. What's going to happen when the next round of replication occurs? Now that's a mutation. That may introduce a mutation over time. And if it's in a gene coding region, it could cause a cancerous mutation, right? As an example. All right? So that error allows your cancer causing errors to accumulate in the DNA. So there's your, so cigarette smoke, pollution. We expose our bodies and DNA to carcinogens every day. The rug you're sitting on, the air you breathe, the water you drink, the UV radiation you're getting from your MacBook, the cellular radiation you're getting from your phone, et cetera, et cetera, et cetera. You are constantly being inundated with problems.
Like when you said like when you get a sunburn or whatever, then the little things fix the DNA. Is that like why people say like if you get 7 sunburns that like you're gonna get like skin cancer because you're not repairing
That's kind of an old wives' tale. Not going to say that you're going to get sick, but further prolonged exposure to UV radiation over time, that increases your chances of one of these or multiples of these thymine dimers not being recognized and repaired. Then it increases your chances for mutation, increases your chances for developing skin cancer. Okay? So, kind of an old wives' tale, but rooted in some truth. Okay? So, we have mismatch repair, which will recognize a difference in A to C, difference in the base pairs, that it should be straight. So it should be A to T, C to G, but if it's G to T, we can recognize that and change it. Another way of doing this is what we call nucleotide excision repair, whereby a nucleotide will be the phosphate diester backbone, that phosphate sugar backbone, will be cut by a nuclease, a protein. It's going to cut it out, remove that nucleotide, and replace it with the right one. All right, so we have mismatch repair. It's going to recognize the mismatch, and we'll remove it and replace it. And we have nucleotide excision repair that's going to cut a piece out and replace it from there. The difference is the nucleotide excision repair is a larger chunk of DNA. Instead of just one base pair, it's a few, 5 to 10 nucleotides. Okay? So here you're showing 2 mismatches. Okay, we actually have 2 mismatch, causes a bulge in the DNA, a change in the diameter. That's a signal for recognition that there's something wrong there. Protein, nuclease binds to that and will cut out this stretch of DNA. That stretch of DNA is then open, leaves a single-stranded stretch. What is right here then? At that, what's going to be right there? We're going to remove this stretch of DNA right here, cut it out. We're going to end up with this, like it looks like this, right? This is 5 prime, 3 prime. This is 3 prime, 5 prime, right? What's right here? You're going to have a free 3 prime OH right at the end of that left side sequence. That is a point where DNA polymerase can bind and add new nucleotides. Polymerase comes in, it answers the iPhone, and we'll start to synthesize the DNA strand and fill in the gap. It's going to fill in the gap. Right? It's going to run into that last. What's going to come in? DNA ligase will connect those 2 strands. We'll bridge that gap. So now it's a continuous strand, and you fix the problem, and now you have the right complementary base pairs, the right form, the right structure to pass on to the next generation. Questions on nucleotide excision repair. So it removes a chunk and replaces it. Mismatch repair removes 1 nucleotide. Now, what does that do in terms of evolution? What does that mean in terms of what happens to changes in DNA over time? If our error rate in DNA is going to be very low, what are the chances that genes are going to change very quickly over time? Low, right? There's not going to be a high incidence of mutation, so it's only going to happen not often, pretty seldom, okay? Very seldom. So, but it does occur. And if it does occur and it's advantageous or coincident with survival and increasing reproductive rate, it will be passed on to the next generation, right? So those changes can become permanent and can drive. This is the part of the driving force of evolution, all right? The mutations that do occur, right? So is associated with, all right? One other problem with DNA replication is that it tends to, there's a problem with replicating the end of the DNA. Now, for bacterial DNA, no problem. Why? It's a circle. Okay? With eukaryotic DNA, it's not a circular chromosome, so we have to get to the end. The reason why the usual replication machinery doesn't complete the last part because of the way it does. On the lagging strand, it doesn't complete the last part, the last so many nucleotides. So if it doesn't complete the last part, over time, what's going to happen? So if I have one round of replication and it leaves off the last 5 nucleotides, right, and then say that gets clipped off, it leaves it. Now the next round of replication is also going to leave off the last 5 nucleotides. And every round of replication that subsequent, it's going to get 5 nucleotides shorter every time. Right? Is that good or not good? Eventually, you're going to get short enough that part of that loss of that DNA at the end is going to be in the middle of a gene. And that's going to be lose a gene, lose function, cause problems. All right? So, over time, you end up with uneven ends. Okay? So it ends up looking something like this. Oh, no, like this. Right? When you have base pairs, and then these aren't filled in. Okay? So, and this is typically on the 3' to 5' end. Okay? Because DNA polymerase would come in on the 3' end and fill out the gap. Okay? So there's another enzyme that comes in and fills in that gap. The ends of chromosomes are called telomeres. All right? Ends of chromosomes are telomeres, and at the ends of chromosomes, there's an enzyme called telomerase, which will fill in the gap. It will fill in and make that run, right? And the action of telomerase is associated with aging and cancer, okay? So over time, your ability to fill in those gaps is reduced. And so over time, your chromosomes do get shorter. And that's associated with old age and losing function, etc. In cancer cells, telomerase is typically overactive. So the cells never get shorter. They always are at its full potential. So they are quote unquote anti-aging type activity. We're not going to. Right, so here's showing, this is a metaphase spread of chromosomes, all right, in a normal human cell, and the researchers in this case have identified and shown where telomerase is active on the chromosomes. Where is it active? Is it in the middle in the centromere? No, it's at each end of the chromosome. All right, so during replication, it fills in the gaps at the end. Right? So that's telomeres. They help prevent the shortening of DNA, right? And then the shortening of DNA is connected to aging. That's something we learned from that. So if chromosomes of germ cells became shorter in every cell cycle, essentially you would end up with less genes over time. So your telomerase catalyzes the lengthening of telomeres of germ cells and in cancer cells and in your cells as well. Okay? Usually it's not too active in most somatic cells, so your cells have a certain lifespan, but it is in your stem cells, right? And then it's inappropriately active, overactive in cancer cells, right? And it has been a large source or subject of research. Okay? The last part of this chapter is how DNA is packaged into the cell. So we've gone over how DNA is replicated and its structure, all right, but we need to understand how a large amount of DNA is packaged into a very small cell, all right. Roughly, if I was to take DNA and stretch it out of the cell, link all the chromosomes together, 46 chromosomes, and spread it out, it would be roughly about 2 meters in length. So about from tip of my middle finger to middle finger. Okay? How am I going to get that linear piece of DNA structured and packaged into a cell that is a cell nucleus that is about 100 microns in diameter that you can't see with your naked eye? All right? And it has everything to do with how the DNA is packaged. We know that a chromosome is made up of what? Lipids? Question mark. Christina? Not lipids. What is a chromosome made of? Protein and DNA. Okay? So we know that in some way, proteins are going to package DNA in a certain way. All right? And that's what happens in a normal interphase nucleus. In an interphase, DNA is not packaged in a chromosome. It's packaged in another way. Okay? But it is packaged in a DNA protein complex. All right? So we have linear DNA and a large amount of it. Right? Bacteria, we have supercoiled DNA, and it is in the nucleoli. In the nucleus, DNA is packaged in the form of chromatin. Okay? Chromatin is different than chromosomes. It is composed of DNA and protein, different types of protein. Okay? Now, all those chromosomes and all that chromatin is fit into the nucleus in a very distinct pattern of organization, organized packing. Okay? So, we have chromatin, which undergoes coiling. So we're going to go from double helix to a coiled helix to beads on a string to a super helical structure, eventually into a chromosome. And it looks like this. So here's your DNA, your double-stranded DNA, right? And the way that we're going to package this together is we're going to wrap that double-stranded DNA around a protein, okay? Well, a complex of proteins, right? What we call histones. So a histone is really a conglomerate. It's a tetramer. It's a complex of 4 unique proteins. Okay? So we have what's called H2A, H2B, H3, and H4. Those are your histone proteins. Okay? They come together, and each of the proteins has these things hanging off it called histone tails, which are important. All right? So the histone is going to come together, and the DNA double helix is going to be wrapped around it. Like that. That's not the best drawing. But it is wrapped and coiled around the whole histone. Okay? So now, what I end up going to, as I wrap that around, I've reduced the 6 feet of DNA to about 12 inches by wrapping it around the histones. And then you look at it here, it shows you your beads on a string right here. See this? Looks like a kind of beads on a string formation. That's actually DNA that's wrapped around the nucleosome, and then a portion of it, of the DNA, then separates between the next nucleosome, which then the DNA is wrapped around again. Okay? So you continue to wrap it around, you now have a beads on a string formation. There's one other histone that they mention called the histone H1. It's called a linker histone, and it helps to direct how the DNA comes into and is wrapped around and exits the histone itself. Okay? Histone proteins that is wrapped by DNA is called a nucleosome. So this whole thing right here is a nucleosome. Okay, so now I have that beads on a string formation. How am I going to pull it in even further? If I orientate the beads in such a way that they are close to one another, I can pack them in a coil, right? And that's the next order of packing. So I'm going to take this 10 nanometer diameter beads on a string formation, and I'm going to orientate them so one's here, one's here, one's here, one's there, one's there. And then I'm going to start putting them on top of one another. So if I was to side view, it would look like this, and then they'd start kind of going in this direction. Okay? Kind of swirling up. Now I've taken 1, 2, 3, 4, 5, 6, 7, say 12 of those, which were this long, and now I've packed them into this long. So taking that now 1 foot of DNA on beads on the string to less than an inch by packing them together. Okay? That 30 nanometer fiber now loops out. So here, the next part is going to show you that now loops out. All right, so parts of it will be stuck together and parts of it will be looped out, giving you what looks like kind of like a pipe cleaner or a bottle brush configuration. Right, so a pipe cleaner kind of looks like this, the middle, right? And then outside of that you have this, I don't know where it went. Anyways, you have the middle metal part, right? And then around that you have the folding. Right, the blue being that 30 nanometer fiber that's looping out and connecting back to the middle. Now I've gone from 1 inch to about 1,000 microns. Okay, now I'm gone from 6 feet into about 1,000 microns of DNA. Okay, and now that bottle brush formation is packed together in what you see a chromosome. That bottle brush formation is your 300 nanometer fiber. I've gone now another 10-fold reduction in size. All right, so now you end up with your chromosome or chromatid that looks like that. Okay, and you see here in this picture, you have the scaffold and your loops, right? Each one of these little loops is a 30 nanometer fiber that extends out. Those are your loops, giving your bottle brush formation. Okay? So we've gone from double helix, wrapping the double helix around the nucleosome to form that 10 nanometer fiber that beads on a string formation, forming the nucleosome. Okay? Arranging the nucleosomes in such a way that we have a 30 nanometer fiber, and then taking that 30 nanometer fiber and then looping it in a scaffolding protein form to form chromatin. Okay? Now chromatin exists in 2 different parts, in 2 different areas. Well, we'll say 2 different categories. It's either really, really condensed or not so condensed. The really, really condensed is heterochromatin. It is not, it's really condensed, it's not open to other proteins coming in. Okay, it's kind of stuck and hibernating. It is not transcriptionally active because in order for transcription to occur, proteins have to get in there, like RNA polymerase and so on. We're going to go through that next week. Okay? Some of it is more dispersed, is more open. It is transcriptionally active. That is euchromatin. Okay? It is open, active. Okay? Genes that are turned on are found in the euchromatin. This is one way, this is the first level of regulating which genes are turned on and which genes are turned off. Okay? Example, we have probably, 75% women in here. What happens, what genetically separates women from men? Or men from women? Which is the Y chromosome or the X chromosome. So, if I, as a male, genetic male, have one X chromosome, how many X chromosome copies do you as females need, or us as humans do we need to have? One. So what do you females do with your extra X chromosome? It's turned off. That is an example of euchromatin, or heterochromatin. Condensed X chromosome, not transcriptionally active. It's turned off. Okay? It's still there. It's not going anywhere. You need it to replicate you need that DNA to replicate when you make more cells or when you produce gametes. It's not going anywhere. But it's not transcriptionally active. It's off. Okay? Think about what separates a stem cell from a regular cell. Is the type of genes that are on or off. And a lot of times, the genes that make a stem cell are turned off and kept in that heterochromatin structure so that it can be passed on, but it's not, those genes aren't on that are found within them. All right, so dense packing make the chromatin largely inaccessible. The chromosomes are dynamic in structure. Okay? These chromosomes and the heterochromatin and the euchromatin, right? They have to have ways of getting open. Okay? And the histone tails, those are a subject of modification. Excuse me. All right? So here we have a histone tail. Sticks out like this. Right? And DNA wraps around it. Okay? Whatever. But they kind of stick out. There has to be times when my cell needs the gene that is wrapped around that histone to be turned on. Right? There has to be times where I need to replicate that DNA. The histone's in my way. I need to get rid of it. So there's a way for the cell to determine and change whether or not the histone's bound to DNA or not. And the tails are the area where that occurs. So if you modify the tail in such a way, okay, that it loosens the association with DNA, then DNA comes off and the histone is separate. Okay? There's also other modifications that make that association more tight. Okay? Depending upon the modification will change the behavior. Okay? That is another way of regulating which genes can be turned on or turned off. All right? Though that's the first level of regulating gene expression, transcriptional regulation. We're going to regulate which genes are going to be turned on or turned off. We're going to regulate which genes are going to be transcribed. Okay? Questions on DNA packaging? No? Yes? Maybe? Clear as mud? All right.